Dead bodies will continue to be transformed into the flesh-eating ghouls.
Hi, I'm Spooky Bill, and you're watching Pathophysiology of the Living Dead, and I am watching The Walking Dead on AMC. Fantastic show, one of the best shows on television right now. Actually, the best show on television right now, and you can't argue with me because I'm right, and you know it. Now, it's a great comic, too. I'm not a comic guy, but I love this comic. So this past episode, episode 5, we got a glimpse at the virus that is causing the dead to rise. And, well, let's take a look at it, okay? So in this shot, we've got two viruses, 1, 2, and they're circular in shape. They have in the center, they have, well, it's possible that in the center, this is single-stranded DNA. However, I'd say it's more likely that it is RNA, right? See, if you look closely, you can see the, well, you can see there's only one strand, which usually is RNA. Now, we see that it's enveloped, right? An envelope is pretty much a protective coat around the virus. You see the darker, probably the capsid right here, and then the thicker lipid envelope here. Now, we've already guessed that if a virus were to reanimate the dead, that it would have to be enveloped. Now, this is because viruses need a living host, a living cell, in order to replicate. When somebody is dead, eventually all the cells of their body are going to die, rather quickly, in fact. And this virus is going to need to survive, because if the dead are walking and biting you, it still has to be virulent. It still has to have some potency. So the envelope protects the virus. It keeps it from drying out, from dying. It's not going to make it immortal, because eventually the virus is going to die. But it's going to prolong the life of this virus. Some viruses can live on an inorganic surface for days. Now, looking back at this picture, we've got our viruses, our RNA, or possibly our single-stranded DNA, but like I said, I think it's pretty much RNA. Our envelope. Now, these studs here on these viruses, they are, well, they're protein studs, right? They are programs for the target cell, and the receptors on this is what attaches to the cell. The virus itself is dependent on this envelope with these studs in order to infect a cell. If the virus itself did not have this envelope, it would not be infective, or as infective, because there's always an exception to the case. So I believe it's possible we're looking at one of two types of viruses. Either we're looking at a retrovirus, which is a single-stranded reverse transcriptase RNA virus. Viruses in this group are HIV. But I think it's more likely that we're looking at a negative sense single-stranded RNA virus. The negative sense is just the polarity of the nucleic acids. Viruses in this group are Ebola, the Marburg virus, influenza, measles, mumps, and rabies. So I think it's a high possibility that this is the type of virus we're going to be looking at in this series. Okay, so there's really no intro to this clip. We don't know exactly what this guy is doing, right? What I can tell you is that that was not an electron microscope. We see the two viruses getting close together, and bam. Okay, so we saw the two viruses. Okay, so we saw the two viruses come together here, and I'm not really sure what happened there. If you slow it down, you can see that we're going backwards now. You can see that they're switching segments of their RNA right there. It looks like the virus is mutating, and there's some type of, genetic recombination going on, right? Genes from this virus are coming into genes from this virus, so that would suggest that these are two different viruses. I'm not sure if we're seeing a new virus being formed, if he's trying to kill a virus with another virus, but some type of mutation is happening here, a genetic transcription, and what happens, again, is the protein, there we go, the protein studs on that one virus have gotten, well, bigger and spikier. You know, they look mean now. No real reason for them to, you know, look like that or anything. But it shows us that it is a different virus now. So what, is this a new virus? Is this a mutation of the old virus? You know, what the heck are we looking at? Unfortunately, we won't see what happens with that because, well, the samples are gone. I don't want to spoil any more of this episode for you if you haven't watched it yet, but let's just say that the tissue sample is no longer available. So, for Pathophysiology of the Living Dead, this has been Spooky Bill, and I hope that you stay spooky. Yeah.